Have you ever asked yourself what would happen if you grew a human brain and an animal brain in a lab and fused them together? I did, and turns out they do better than human brains alone. I will explain. Just in case you're not familiar, you can grow a tiny version of a human brain in a lab. This is called a brain organoid. It can be done from skin cells turned into stem cells. There are a wide variety of reasons why you'd want to do this. One of them is computation, because brain organoids can operate as a form of AI. The other one is medicine, because these can be treatments for strokes and have had very good results for mouse models and even simian models. It just hasn't been done in humans yet, and there's good reason for that, because you don't really want to mess with your brain. While we have transplanted them into other animals and had very good results, we still want to know what's going to happen with them when they're grown before we transplant them into a human being. A lot of stem cell research has been done with trying to use stem cells to heal strokes, but it's not very effective. It does better if you grow them into a little brain before moving them. Researchers have run into a lot of barriers when it comes to growing them independently of a brain, because inside of a mouse brain, they do grow. They make projections all the way through the brain. They repair damage. They effectively act as a baby brain. Yes, they are highly immature brain tissue, but because of that, they can integrate and they can mature and become a functional part of a mouse's brain. Of course, there are a lot of ethical implications to this because this is humanizing animals. It's taking human parts and integrating them, and we tend to apply human morality a little bit higher than we do with other so-called lesser animals. Keeping them alive and active and functional is very difficult inside of a petri dish, in part because they don't have a lot to do and our brains need something to do. So why not co-culture them? And yes, that is the idea, lump together a brain organoid with a slice of mature mouse brain. That is exactly what these researchers did, and it turns out they live longer and do better. These were human microglial cells. This might seem like a slightly strange idea because microglial cells are part of your brain's immune system, and while your brain doesn't really have that aggressive an immune system, you really wouldn't want it to, it does have it, and that makes up about 10% of your brain's total cells. They integrate just fine with something that is not human, although that is not really the point of this study. They're not just trying to see if we can make chimeras. They're trying to observe brains over the long term. Having more mature brains gives us a platform to study human beings in something that is not a live human being. I don't know a good way to say this other than brains just need something to do in order to remain functional. That does make sense too, especially if you think about human beings. We need to use our muscles or they'll atrophy. Our brains also need to be used in order to create neural pathways and maintain them. Otherwise you will forget, and it is a reason that I don't know how to do long division. Theoretically, I knew at some point and I could probably learn it again, but I also have no reason to do so. A lot of people find this research generally creepy, and I do understand this because the brain is so integral to what we are. If there ever was a soul, it would probably be present in the brain. The idea of taking something that makes us human and transplanting it into an animal should make you uncomfortable, and maybe so when it comes to growing parts of humans in a laboratory. This is one of the reasons that researchers are hesitant to call brain organoids sentient or conscious. Well, yes, some researchers have claimed sentience. If the public has a major backlash against this research, it could hamper it for decades to come. We don't understand sentience or consciousness enough to even really apply it to anything. We understand our own experiences, ideally inside our own heads, but we cannot apply that to others because we do not know what their experience is like. If we start applying this to neuronal cultures in a dish, then, well, we're being dishonest, to be fair. Instead, I propose a middle ground. We cannot make claims that we just can't defend, so we're not able to say that these things are definitely conscious, and we're also not able to say that they will never be conscious, especially when we are lobbing them together, embodying them in video games and robots and other animals. We can make them larger. We can make them live longer. Should that research necessarily be done? I'm not sure. I really do think it should be investigated as a treatment for strokes. I'm not wild about biological neural networks as a replacement for computation with AI. It just feels wrong, and I know I have no good reason to say that, so it's a good thing I'm not in charge. I really hope you've enjoyed this. I will keep you up to date. You have no choice. Follow for more.